Now today my topic is C4 cycle. This is called as Hatch-Slack cycle. So C4 cycle, Kochak and Burr, these are two scientists in 1965 identified that in some plants growing in tropical regions, the first stable compounds formed in a dark reactions are C4 dicarboxylic acids like oxaloacetic acid or malic acid or aspartic acid. So they conducted experiments in sugarcane using radioactivity. These are carbon dioxide. So the first C4 cycle was identified by Kottachak and Burr in 1965. So they identified in tropical region plants and first stable compounds formed in dark reactions and they carbon C4 dicarboxylic acid like oxalic acid and malic acids and aspartic acids. They conducted experiments in sugarcane. This is the called as C4. Later Hutch and Hutch scientist, his name was Hutch and C.R. Slack. C.R. Slack in 1966. They are indicated that C4 compounds are fast formed stable compounds in the photosynthesis of sugarcane leaves and that C4 pathway operates an alternative to C3 pathway. So C4 pathway is reported in 19 genera and 90 species belonging to 11 families of angiosperms. So it is the identified in millets like jowar, maize, and per millets, um, finger millets, pennisetum, um, typhodium of the family of Poaceae. These are almost Poaceae. So, and in members of um, cyperaceae plants, in dicotyledons belonging to the families like Amaranthaceae, Portulaceae, uh, Boraniaceae, and so etc. So almost the Professor V.S. Ramdas, internationally reputed Indian. He was the Indian scientist, and C4 pathway in the weed plants of Tirumala hills like Tribulus terrestris and Euphorbia heta and Amaranthus spinosus of Amaranthus. All the characters with which C4 plants can be identified, it is called as C4 syndrome. So this is the leaf anatomy of C4 plants. So in C4 plants, through belonging to derived families, and these are the taxonomically unrelated. These are common features in the leaf anatomy. All these plants possess bundle sheath made of large sized cells. The bundle sheath, this bundle sheath This bundle sheath made up of large sized cells. The bundle sheath resembles flower wreath. It is called as Kranj anatomy. German Kranj flower wreath. So the chloroplast of mesophils and bundle sheath differ in structure and functions. So mesophils chloroplasts are normal in the size with grana. These are the mesophil cells. So these are the normal like grana with grana. Both photosynthesis 1, photosynthesis 2, PS1, PS2 are active in them. So PEP carboxylase is more active in the Kranj C4 plants. In bundle sheath, uh, this bundle sheath chloroplasts are bigger in size. Agranal with thylakoids arranged separately, not forming grana. Only PS1 is active in them. RUBP carboxylase is more active in them. So mesophil chloroplasts, these are specialized for primary carboxylations. The bundle sheath chloroplasts are specialized for carbohydrate synthesis. So this type of dimorphism of chloroplast with division of labor is exhibited by all C4 plants. In C4 cycle, basically there are 4 phases. Formation of C4 acids with the help of PEP carboxylase enzyme in mesophils, and transport of C4 acids into bundle sheath, decarboxylation of C4 acids to release carbon dioxide, which is fixed as carbohydrate by C3 cycle. C3 acid formed in a decarboxylation moves back to mesophil and help in PEP generation. So PEP carboxylase catalyzes primary carboxylations. The enzyme is concentrated in cytoplasm and use HCO3 as substrate. Carbonic anhydrase enzyme enables the formation of carbonic acids from carbon dioxide and water in the less than 1 second and form carbonic acids. So these reactions of C4 plants are explained by Hatch and Slack, and so their cycle is also called Hatch-Slack cycle. The reactions of C4 cycles are indicated so phosphophenol pyruvate plus HCO3 pepcase give rise to oxaloacetate plus HOPO3. Phosphophenol pyruvate acts as carbon acceptor and utilizes HCO3 in presence of carboxylation occurring beta carbon of PEP. PEP case it is a B carboxylation. PEP case have more affinity of carbon dioxide than that of RUBP gas, and its activity also is very more. Oxaloacetate plus NADPH plus H plus, these are gives rise to malate plus NADP. Oxaloacetic oxaloacid, which is a C4 dicarboxylic acid, formed is reduced to malic acid in presence of NADPH, formed by light phase NADP malate dehydrogenase acts as a catalyst. So this is almost this is the leaf anatomy of C4 plants. Third reaction is the oxaloacetate plus glutamase give rise to aspartate plus alpha ketoglutarate. In some plants, oxaloacetate is transmitted to aspartate in presence of oxaloacetate aspartate aminotransferase. So malate plus NADP plus pyruvate plus carbon dioxide plus NADP plus hydrogen. Malate formed in the mesophyll moves into bundle sheath and bundle sheath and in presence of malic enzyme from pyruvate. So and carbon dioxides. Malate acts as a carrier of both carbon dioxide and reductions power in the form of NADPH. As bundle sheath chloroplasts lack PS2, they are unable to synthesize NADPH and it must be provided from mesophils. Pyruvate plus HOPO3 plus ATP give rise to phosphopenol pyruvate. Pyruvate formed in bundle sheath is transported to mesophyll and in presence of ATP utilizes to phosphate bond energy and helps into the regeneration of carbon dioxide accept EP. So the carbon dioxide released bundle sheath, it is used for carboxylation of RUBP and in presence of the enzymes Rubisco, hexosynthesis takes place through Calvin cycle in C4 plants along with C4 cycle, C3 cycle also operates. Bundle sheath cells and mesophyll cells exist in the close associations. The bundle sheath and mesophyll cells, these are the close association with each other. They are also connected by plasmadesmata. The leaves with Cranch anatomy can be recognized with the naked eye due to their dark green veins. In C3 plants, the veins in the leaves appear pale due to lack of chloroplast in the bundle sheath. Primary, the carboxylation takes place in mesophyll and secondary carboxylation in bundle sheath. In carbon dioxide released from C4 compounds is used for RUBP carboxylation. As carbon dioxide is available in higher concentration to Rubisco, the scope for photorespiration will not exist and thus photo respiration is negligible or absent in the C4 plants. Further in the double shade, chloroplast pH2 is absent and oxygen is not released. This also helps rubisco to act only as carboxylase and photorespiration is absent. When oxygen concentration in atmosphere 21% oxygen concentration in atmosphere, it is reduced to 2%, and the rate of photosynthesis is not affected in them. 2% of oxygen reduce photorespiration, and thus rate of photosynthesis increases in C3 plants. Carbon dioxide compensation points is less in C4 plants. The concentration of carbon dioxide at which the rate of respiration and the rate of photosynthesis the equal is called as carbon dioxide compensation point. In C3 plants, carbon dioxide compensation point is more 40 to 60 ppm carbon dioxides at compensation points, and the net gain of photosynthesis is 0 in C4 plants, and this net photosynthetic rate is more and growth is more, but unfortunately most of the C4 plants are wheat plants. So trials these are progress to incorporate C4 cycle in C3 crop plants. Types of C4 plants, these are three different types of C4 plants are recognized. The primary carboxylation is similar in all of them, but depending on the C4 acid that is transported from mesophyll to bundle sheath, nature of enzyme involved in decarboxylation reactions in bundle sheath cells, and their intercellular localization differences are recognized. First one is NADP me type, second one is NAD me type, third one is PEP CK type. So NADP me type, sugarcane, maize for example. So NAD me type example amaranthus. So PEP these are chloris and panica. So NADP me, these are mallet formers. So the first in these plants first oxaloacetic acid and then malic acid is formed, and it moves to bundle sheath chloroplast. There it undergoes oxidative decarboxylation to form a carbon dioxide, and pyruvate formed moves back to mesophils and converted to PEP. So this is the NADP. So an NADP as coenzyme, and that these plants are called as NADP plants. NAD me type, these are aspartate formers. In these plants, malic acid is converted to aspartic acid, and mitochondria of bundle sheath cells undergoes transamination to produce oxaloacetic acid. Oxaloacetic acid is reduced to malic acid in the chloroplasts. Malic acid releases carbon dioxide, and pyruvate is formed with the help of malic enzymes with NAD as coenzyme, and so pyruvate is converted to alanine by elimination, alanine enters mesophyll by dimension converted to pyruvic acid. Pyruvic acid is converted to PEP. So PEP in presence of pyruvic dikinase. Next PEP CK type. So this is the phosphophenol pyruvic carboxy kinase type. So these are also called as aspartate formers. However, aspartic acid entering bundle sheath is converted to oxaloacetic acetate and instead of being reduced as in NAD me type, so releases carbon dioxide in the chloroplast is presence of ATP and PEP carboxykinase enzyme. So carbon dioxide is used in the carbohydrates synthesis by Calvin cycle. Pyruvic acid is converted to alanine and enters mesophyll as the enzyme involved in the decarboxylation is PEP carboxykinase. So these plants are called as PCK type. In C4 plants, as both of the pathways C3 and C4 operates in the same plants, the scope of efficient usage of carbon dioxide is more. It acts as adaptation in plants growing in high temperature and drought conditions. In enable the plants grow efficiency in the those high temperature and the water scarcity. The studies of C4 pathway by the way for the concept of getting high yield by suitable modification in plants. Further, the understanding of C4 plants may help in selection of plants with efficient photosynthesis. So this is the C4 C4 cycle, Hatch's cycle. So